Hello everyone, I'm Roshni Gandhi from Dr. Circe's lab, and today I wanted to share some findings from my summer project on improving penetration of immune cells into solid tumors through the use of polyethyleneamine-coated microbubbles. I would like to start off with an introduction to neuroblastoma, which is a type of cancer that is found in children and infants, and it's most commonly found in the adrenal glands. It's also the most common solid extracranial tumor in children and infants, and it affects every 1 in 8,000 live births. Although survival rate and impacts on health vary depending on many factors, treatment for high risk neuroblastoma requires intense chemo and radiotherapy, which causes severe short and long term effects on health, some of which include fatigue, heart problems, and bone and joint problems. And there are current strategies being used to treat neuroblastoma, one of which is immunotherapy, which uses the body's own immune system to fight diseases. And it's quickly becoming a frontline treatment option due to its many benefits, one of which includes the ability for the immune system to recognize and target cancer cells. However, there is a major limitation of immunotherapy where poor penetration of leukocytes to the solid tumor occur. The goal of this study is to create an immunotherapy technique where leukocytes are directed to the site of the solid tumor to attack cancer cells. And now I'd like to go over some information on ultrasound contrast agents or microbubbles, which are used for imaging and drug delivery purposes because of their ability to remain in circulation and their response to radiation force application. As you can see in figure 1, microbubbles have an outer shell made of lipids, polymers, or proteins, and they have a gas core. And primary radiation force application uses ultrasound waves to displace microbubbles to adhere them to the vessel wall, as you can see in figure 2. So this is a novel strategy of loading polymer onto lipid microbubbles and using them to direct cells. And microbubbles have not been bound to cells before for this type of application. So the first step is to make polyethyleneamine-coated microbubbles. And PEI is a well-known cationic polymer, and it can be used to improve delivery of cells to the site of the tumor because of its branched structure. So the first step was to make DBCO microbubbles, which is a type of lipid microbubble, and then mix them into a polyethyleneamine stock solution. So the polymer attaches to the lipid microbubble because the azide and DBCO functional groups interact in a click chemistry reaction, and this is a one-step fast reaction. And then I wanted to test the polymer attachment onto the microbubbles. So in order to do this, I used a fluorescentamine assay where fluorescetamine is a reagent that detects primary amines. I tested this assay and I was able to determine that for DBCO microbubbles, there was more loading of polymer when compared to regular lipid microbubbles. But however, due to the variability of the study, this result was not statistically significant. Therefore, I decided to gather qualitative data on microbubble attachment to cells. And as you can see in figure 4A, these are unbound neuroblastoma cells. And in figure 4B, these are polymer loaded microbubbles that are attaching to neuroblastoma cells. And the black circular dots are the polymer loaded microbubbles. And this occurs because of electrostatic interactions where the polymer is positively charged and the neuroblastoma cell is negatively charged. And so the next step was to demonstrate the use of ultrasound radiation force for directing microbubbles. As you can see in figure 6, this is a setup of a microscope chamber, and I pushed microbubbles through a capillary tube and then applied ultrasound radiation force. In figure 7, for no flow conditions, this shows primary radiation force application and how it moves microbubbles bound to cells to the capillary tube wall. And in figure 8, it shows inflow conditions where microbubbles attach to the capillary tube wall and catch neuroblastoma cells. And for this, a 0.18 megapascal pressure amplitude was used. Over the course of the summer, I was able to make PEI loaded microbubbles and successfully direct cells to capillary tube walls through ultrasound radiation force. This study was an initial model using the resources available to show microbubble binding and primary radiation force application. The attachment of leukocytes uses the same concept of electrostatic interactions, and this can be further tested in future experiments. Thank you so much for listening.